Good morning, everyone, to Grand Rounds. This morning we have what we call our translational Grand Rounds, which is just, simply a word to say, we're bringing different insights from the, the clinical and the basic and the translational world together. And I'm very, very happy today to introduce my colleague, Dr. Lars Maegdefessel from Munich. So Dr. Maegdefessel and we met, we were probably postdocs. He was a postdoc at Stanford after doing his training as clinical and his doctoral training in Germany. He left Stanford and he went to the Karolinska, where he established a really incredible research program studying atherosclerosis and aortic diseases. And again, really at that translational interface. He then actually took on a position as professor of vascular biology at the Technical University of Munich, or TUM, in 2023. And since then, he's been sort of, in two places at once, both in Munich and in Karolinska. I was actually lucky enough to recruit a technician from his lab, which who benefits my lab greatly, Nancy. So we're very, very glad to have her, and then trained by Lars. So Lars's interests and where we sort of also met each other a long time ago was in the field of non-coding RNA and how those regulate sort of smooth muscle cell phenotype and plasticity in the vessel wall. And now he's really gone on to form not only a research program examining these, regulatory mechanisms, but also establishing a really vast biobank, which I'm sure we're going to hear about today. He did this at the Karolinska, actually, in the Department of Surgery, where they essentially treated, every sample, every patient as, a research subject, and every piece of tissue and every sample that they were able to collect went into their biobank. And they since have made these international collections with their, what they call the bike biobank. And he's been able to establish that in Munich also. So today we're going to hear about the multi-omics approaches for human atherosclerosis. And afterwards we'll have Q&A on both online and in person. So welcome, Dr. Maegdefessel. Dr.
Yeah, thank you, Katie, for this very kind introduction. Everybody for being here this early. For me, it's fine. It's 1:30, so perfect time to give a talk. As mentioned, so I'll mainly talk about this kind of multi-omics approach and how we try to kind of decipher mechanisms in human atherosclerosis. And as mentioned, that's something. When we started doing this, I thought we would be doing this for maybe 1 to maybe 3 years to get some cool targets and then take that to the wet lab. But it feels like we've not been doing anything or much else the last 10 years, I have to say. So it's really been quite a journey. And finally, I think we have something we can kind of show and put forward of how we believe this multi-omics kind of approach helps us to really decipher what's driving atherosclerosis. And when I speak about atherosclerosis, I mainly speak about carotid artery disease, because there's really some differences between the vascular beds, between the carotids, the coronary system, and also for peripheral artery disease. So for us, as we have fairly easy access through vascular surgery to these carotid specimen, it's something where the biobank really is much focused on carotid artery disease and stroke, and not so much on the other vascular beds. So everything I'll talk about today, when I speak about atherosclerosis, it's in the context of carotid artery disease. And stroke, obviously, I don't really have to mention that to this audience. A lot of clinicians here, so you might know this disease much better than I do. And not to scare anybody, but 1 out of 4 people will suffer from stroke during their lifetime. It's really become a big issue, especially in low to middle income countries, where stroke has kind of overrun or overturned myocardial infarction as the most common cause of death. So in most of these countries are very populated, like China, India, sub-Saharan Africa, where stroke has really become probably the major health problem. What's also kind of interesting is that up to roughly 80% of strokes occur in people with low to moderate cardiovascular risk. So there's something about the disease. It's usually not the 80-90% stenosis that ruptures. It's usually the 50, 65 to 70% stenosis that becomes unstable, ruptures, and can create a stroke. And of course, there's many other reasons that can also lead to stroke that are not mentioned here apart from carotid artery disease. And of course, it also creates a huge health burden. It's actually the most costly disease we currently have, or we're spending roughly 450 billion US dollars, which is roughly 0.4% of the global GDP. So again, creating really a major burden on our healthcare systems. And as mentioned, so what we do, we've been in Stockholm and also in Munich collaborating very closely with our vascular surgery clinic. So I'm a cardiologist by training. I'm not a vascular surgeon, but that was always something that kind of helped us to really be truly translational and very close to the disease, is to work and to implement studies on these human biobanks. And there's this kind of window of opportunity that opens up once you get these carotid and arterectory specimen. And these you can characterize, and these you can study. And we've been doing this, as Katie mentioned, for quite some time now. And this is kind of how now the Munich Vascular Biobank is set up. So this is what we are doing with our samples that we have in Munich. And we have over 4,000 carotid and arterectomies that we are currently working with, or we have samples in the biobank. So we do a lot of, we try to genotype all of our patients. We've been more recently also trying to do a lot of looking into clonal hematopoiesis, so chip calling on these patient specimen, to have that kind of like as a risk factor in our biobank as well. And there's a couple of collaborations that are looking in particular in chip in atherosclerosis that we are involved in. We'll do, and I'll show quite some examples of how we use different transcriptomics methodologies, bulk sequencing, which is something we still like to do because you can still do a lot of samples and go fairly deeply on RNA expression using bulk RNA sequencing. Obviously, single-cell transcriptomics, and more recently also spatial transcriptomics, which we don't really use for discovery. We mainly use this as an integrative approach with the bulk and the single-cell sequencing to more or less visualize whatever we are seeing, mainly with single-cell sequencing, also in using the spatial technologies, because location really does matter in atherosclerosis. And again, I'll show some examples of that. And then more recently, we've also been just very lucky to have Matthias Mann, who's one of the godfathers of mass spectrometry research, very close by, just down the road. And we've been starting to do a lot of proteomic studies, in particular focus on spatial proteomics, so doing single cell, deep visual proteomics to, again, kind of in a spatial context, decipher what proteins are different and are changing during the context of carotid artery disease and plaque vulnerability. We are integrating a lot of the patient metadata, so the clinical data bank, if you wish, is directly integrated with a biobank. So we kind of collect over 30 different clinical variables. So we look, of course, at patient symptoms. We have all the risk factors, medical therapy, history of the patient, and other concomitant diseases. We've been also more recently trying to use medical imaging a lot more, so CT and MRI imaging, which we are trying to especially overlay with the spatial transcriptomics to give us some hints and some clues of how we probably don't even have to analyze the plaque itself, but can use imaging to determine signals that kind of tell us which patients are more likely to develop a vulnerable plaque and more likely are requiring surgery. And then histopathology, of course, plays a huge role. So again, we fully characterized all of the tissue samples we are getting. And we use different criteria. One of them is the so-called Oxford criteria, which mainly looks at the thickness of the fibrous cap, but also the AHA criteria, which uses kind of judgment on size of the necrotic core, if there's intraplaque bleeding, and other things also plays a huge role. And again, in particular, in the context of kind of submerging spatial transcriptomics with histopathology and using machine learning to really kind of decipher if there's certain patterns within a plaque that are more commonly occurring in an unstable lesion or in a symptomatic patient. So the first part, I, as mentioned, introduced you mainly to the transcriptomic approaches we've been using. So again, we've been doing a lot of bulk sequencing on roughly 500 pairs of samples so far. And what we've been trying to do a little bit different compared to some of the other biobanks is that we are trying to always kind of compare the most advanced part of a plaque, so this most advanced part of a lesion, to this more or less adjacent, more proximally sitting control piece. But it's not really a control piece, as you can imagine, because everybody in this room has atherosclerosis, so it's more a piece that shows signs of early lesion development, so internal hyperplasia, some lipid deposition. And we are always kind of comparing that in a paired manner, so trying to capture this dynamic of what's going on from this early lesion, here mentioned as a control, but again, not truly a control, towards this most advanced piece of the plaque. And that's something we've also been doing in 32 pairs now, using single cell sequencing. And as you can imagine, all the different subclusters or cell states that you're expecting are, of course, there. Single cell sequencing, of course, a very nice, super cool technology, but a little bit biased towards immune cells, which is giving you some headaches and some problems if you're also interested in the stromal cell compartment, so smooth muscle cells and endothelial cells. And the reason mainly is, so smooth muscle cells, fibroblasts, also endothelial cells are more, they kind of need adhesion to a neighboring cell. They usually sit in the matrix, and with all the protocols that you need to get your single cell suspension, a lot of these cells actually die off. Immune cells are more happy with these procedures. So there's always a certain immune cell bias that you are seeing with single cell sequencing, which if you are in the immune cell field, it doesn't bother you that much, but it doesn't really give you, of course, reliable information on the position of these plaques. Luckily, there's a lot of different bioinformatic tools that you can use to kind of deconvolute RNA sequencing, bulk sequencing into single cell sequencing. One of them is mentioned here by Prism, but there's also others like Harmony or Music that you can use for this, where you basically use the cell labels, the cell state labels that you have from single cell sequencing and then deconvolute your bulk sequencing data. And again, in our case, we have 500 plus bulk sequencing data. So it gives you a more realistic picture of the proportions of the different cell types that we are seeing in our human plaques. And this is kind of the changes on a relative expression level that you're seeing for all the different cell substates. And what's been quite interesting for us to see if you do this deconvolution of your bulk stack data into the single cell data, the most abundant cell types that kind of all of a sudden surface are different smooth muscle cells. Or smooth muscle cell substates, and that's something we've been interested in for a long time, trying to understand that plasticity in smooth muscle cells and how they are adapting, how they're changing during plaque progression, and how they potentially also contribute to lesion instability. And roughly 65 to 80% of all the cells that we find in plaques, at least based on this deconvolution approach, are different states of smooth muscle cells that I'll talk about in a minute. But of course, also immune cells, super important. So most of the macrophage substates, also T cells, B cells, we are less prominent with this approach. They double and triple in number, if you wish, in these more advanced plaque pieces compared to early lesions. So they're almost not present at all in early lesion development, so during the more or less atherogenic process, but they are really very prominently present in these more advanced plaque pieces. But again, so for us, and that's also something to mention because that data, and we know that, and we've been battling with reviewer 2 for quite some time, with several of our papers. Of course, there's a difference in between biobanks and how the samples that we are analyzing are being composed. So this is like, I would love to say a prototypical plaque from our biobank, but of course it's a nice representative image. And what we usually have in our Munich biobank is where our vascular surgeons kind of maintain a lot of the media within these plaques. So of course, our samples might be a little bit richer in smooth muscle cells than some of the other samples that other biobanks, prominent biobanks like AtherOxpress, for example, have been collecting, which mainly, we believe at least, I'm probably analyzing, when they talk about the plaque, they are mainly analyzing the necrotic core and not so much that whole vascular context that we are probably seeing in our carotid artery disease specimen. But again, coming back to the smooth muscle cell plasticity, and that's something, again, that we've been quite interested in over the past years, is trying to understand that plasticity of smooth muscle cells, how they're adapting, and what role they play in the context of atherosclerosis, and then especially plaque destabilization. So these are kind of like the 4 subclusters of fibro smooth muscle cell types that we are seeing. So we see this more fibroblast-like SMC cell type, which is producing a lot of matrix, shows high expression of different collagen genes. We see the classical indicated here in blue contractile smooth muscle cell that mainly resides in the media of these plaques. And then this modulated SMC phenotype that we are very interested in, because it seems to be that there might be some type of switch going on in these modulated SMCs that where it's kind of decided whether these cells become either more inflammatory or more matrix producing. And that concept of plasticity has been described for a very long time. So Martin Bennett, Sanjay Zinia, and then in particular Gary Owens, who's been doing a lot of mouse lineage tracing studies on smooth muscle cells and how they contribute to lesion development and also lesion destabilization, have been shown for quite some time now that you have this classical contractile smooth muscle cell that again resides within the media. They turn into this more modulated phenotype that we are also picking up with single cell sequencing, and then some type of checkpoint decision is being made, whether they become more of a macrophage-like cell, a term that I don't really like because macrophages are much more complex than these smooth muscle cells that we are seeing here, but they become more inflamed. So they have to handle lipids, smooth muscle cells, and that's something they are not good at. And they die off very quickly and release a lot of cytokines, release a lot of inflammatory markers when they're dying off. Out of this more inflamed, more foam cell-like smooth muscle cell phenotype. And that's definitely has been shown many, many times to contribute to lesion destabilization. And the other path that they can take, again, is this going into this direction of a more fibroblast-like cell where they produce a lot of matrix and have been shown to also contribute to lesion stabilization. And again, that kind of crossroad here. So what is happening in this modulated, more intermediate state of a smooth muscle cell, and can we drive them to become more fibroblast-like and less foam cell-like, is a signal that we've been trying to understand and trying to unravel in our lab. And you can look, based on this, you can look at pseudotime analysis based on splicing events in different transcripts. If you look at the top 20 transcripts that seem to be most heavily spliced and potentially involved in this plasticity response in smooth muscle cells, what's been quite interesting for us, and also as Katie mentioned, we have quite some interest in non-coding RNAs, that over half of the transcripts that seem to be involved in this are actually long non-coding RNAs. And that's been quite striking, quite interesting, and we've been looking at a couple of them and what role they play in this disease context. And the other thing, why long non-coding RNAs could also be very interesting in that context and could also be potentially used as therapeutic agents in that context, is because they are very cell type specific. So a lot of these, almost all of these long non-coding RNAs that we are picking up are exclusively expressed only in some of these smooth muscle cell substates. And one of them, this DLX6 antisense 1, is also already has been shown by us and also by others now to be exclusively expressed in this modulated SMC phenotype. And modulation of this again is very interesting, because if you knock it down with antisense oligonucleotides or siRNAs, you can drive cells towards this more fibroblast-like phenotype, and you kind of prevent that transition into a more inflamed smooth muscle cell. I don't want to bore you too much with non-coding RNAs or long non-coding RNAs. Again, they've been shown to be important gene regulators exerting different functions within the nucleus, but also within the cytoplasm. So they can have been shown to act as hosts or sponges for microRNAs. They affect protein and mRNA stability. And they've also been shown to chaperone proteins, shuttle proteins, transcription factors, for example, from the cytoplasm into the nucleus. And again, there's many therapies that are aimed at targeting them, mainly inhibiting them using antisense oligonucleotides or siRNAs. We had more recently 2 studies on these long non-coding RNAs. One is about a long non-coding RNA about MIAT or MIAT. So MIAT stands for myocardial infarction-associated transcript, was initially described as a GWAS hit without really knowing what the biological function of this MIAT transcript is. And what's been interesting is that MIAT is almost exclusively expressed in fibrous caps of human atherosclerotic plaques. And then mechanistic studies in vitro and in vivo have shown that MIAT actually binds to the promoter of the KLF4 gene, which has been shown to be a very important transcription factor in this transition of smooth muscle cells towards this more macrophage, more inflamed phenotype. And if you knock that down with antisense oligonucleotides, you keep smooth muscle cells in their more or less contractile state, contractile phenotype, and can stabilize lesions with this. And then another link RNA that we've also studied in that context of smooth muscle cell plasticity and plaque stabilization, but also in preventing aneurysms, is this link RNA termed in6, which is an antisense transcript to the fibroblast growth factor 2. And fibroblast growth factor 2 behaves like a growth factor, as the name says, in vascular diseases. Again, also very much enriched in smooth muscle cells. And if you target that with ASOs, you can again increase expression of this growth factor of FGF2, and you can stimulate smooth muscle cells to become more fibrotic, more proliferative, which again prevents aneurysm formation in mouse and mini pig models, as well as stabilizes plaques as a potential novel therapy in this context of vascular diseases. The way we are trying to in a fairly translational way, trying to transfer what we are doing in humans to also to mouse models is we've been using a lot this kind of what is termed an inducible plaque rupture model. Because one issue that we are seeing, especially with all the high-fat diet models that we've been doing, that these don't really recapitulate what we are seeing on the human side. So again, location really seems to matter, and processes that are ongoing in the carotid plaque don't necessarily occur in the aorta of a mouse model. So we've been doing this inducible plaque rupture model where you do an incomplete ligation of the carotid artery of these 16-week-old ApoE knockout mice, so pro-atherogenic mouse model. And then after 4 weeks, you place this conical cuff just below this incompletely ligated carotid artery, and that puts tensile stress on these lesions, on these athero lesions, and roughly 50%, as indicated on these images, show signs of ruptures that are very comparable or somewhat comparable to human disease. So they show endothelial cracks, they show that the fibrous cap can thin out, and they show atherothrombotic events as indicated by this cross-linked fibrin antibody. And very similar to humans, we've been doing a lot of single cell and bulk sequencing on these mouse lesions in a very similar setup. So we used the injured, the ligated part as our advanced plaque, advanced lesion, and the contralateral side that you see here, because it's an atherogenic mouse model, more or less as an internal control. And what's been quite interesting when using this mouse model is that we also see here already with the lowest resolution in single cell sequencing that we can also determine and pick up this modulated SMC phenotype, a phenotype, again, that we're not seeing in mouse aortas, but really only in these mouse carotid arteries. And what's also been interesting and very important for translation of the human finding also into these mouse models is that the cluster defining genes for these modulated SMCs are very similar and almost identical to the ones that we are seeing in human disease, including, again, this DLX6 antisense 1, which is a very interesting link RNA, as well as its sense partners, DLX5 and DLX6. So very similar cell type that we pick up here in these mouse models. And again, there's a lot of studies going on trying to modulate now in this mouse model, this modulated SMC phenotype, trying to push smooth muscle cells away from this more inflamed foam cell-like phenotype and to push them towards the direction of a more fibroblast-like matrix-reducing and plaque-stabilizing phenotype. But back to the human studies and back to the multi-omics that we've been doing. So as mentioned, we do a lot of integration of bulk, single cell, and then more recently spatial transcriptomics. Not really to do discovery with spatial transcriptomics, but really much more to use spatial transcriptomics as a tool to delineate and to show, to visualize substates of cells that we are seeing with bulk and single cell sequencing. And we've been using this Xenium panel. So it's a custom-made panel. So when we started using Xenium from 10x, there was, right now they have this 5K panel, so 5,000 genes that you can then, in addition to that, build your own custom panel. In our case, at the time, we could only create our own targeted panels, and we did this by using 547 genes based on our own single cell and bulk sequencing data to create these panels. And what's again been kind of interesting that you can really, as mentioned, kind of visualize and kind of show different cells, cell types and cell substates using this technology. So for example, endothelial cells, which not only line up the lumen of these plaques, but they also kind of, as you can see here, show up in these more angiogenic features. So these are sprouting vasa vasorum that sprout into the plaque, become leaky at some point. And when they become leaky, a lot of immune cells are actually entering the plaque through these kind of neovessels or leaky neovessels, which has been shown by us as well as by others to create plaque destabilization and symptoms in these patients. What's also interesting about the technology is that we pick up cell types that we are missing with single cell sequencing. So neutrophils, for example, we don't see with single cell sequencing. With the spatial transcriptomics, as it's an in situ sequencing, so it's really a snapshot. We all of a sudden see neutrophils, again, in very similar areas where we also have these angiogenic sprouting neovessels. And what spatial transcriptomics also confirmed is that, again, smooth muscle cells being highly abundant in these plaques. And in addition to these more contractile SMCs and the modulated SMCs, that interestingly very similar to how it's been described in this scheme from Gary Owens, really reside in this kind of border zone of the media where these cells kind of become migratory and enter the necrotic core. We see these fibroblast-like SMCs, also interesting because the location, as thought and as kind of expected, is mainly in the fibrous cap structure of an atherosclerotic lesion in humans. And then also this more inflamed phenotype that, again, we're also missing with single cell sequencing. So we don't really see this foam cell inflamed smooth muscle cell phenotype with single cell sequencing. We're missing them. But with the spatial transcriptomics approaches, again, they pop up and they are very prominently distributed throughout these advanced lesions in humans. And just as an example, again, the paper is almost published, at least accepted in principle. And there's an online tool that you can use to look up your favorite cell type and to look up your favorite gene, if it is on that gene panel at least. You can use that again to really spatially locate different cell substates. And for macrophages, that's, for example, been quite interesting because there you see that there seems to be like a certain gradient going from these more like C1q, HMOX1 positive macrophages that kind of reside very close to the lumen, involved in heme degradation, also very close to these areas where we have these intraplaque pleadings, all the way to a more TREM2 high kind of phenotype that really then resides fairly far away from the lumen and more in these shoulder regions. So more in an area where these TREM2 high macrophages seem to be also probably involved not only in foam cell formation, but also in a more pro-fibrotic response. And that's you can show by, again, looking at the distance to the lumen for these different substates of macrophages. And it's something we could also resemble in our single cell sequencing data that shows this potential, at least transdifferentiation from these C1q HMOX1 positive macrophages towards these more TREM2 high macrophages that reside, as mentioned, further away from the lumen and more in the plaque shoulder in an area where a lot of remodeling is actually going on. And we've also been doing, and this is just one example, doing a lot of machine learning of our histopathology and then trying to integrate the signal we are getting from the spatial transcriptomics. And this is just examples of roughly 100, I think 120 plaques that we are getting. And what we've been doing in collaboration with Ming-Yao Li at the University of Pennsylvania is to, in a semi-supervised way, just tell an algorithm whether the plaque the algorithm is seeing and detecting stems from a stable or an unstable lesion. And we've done this for over 1,000 plaques right now on these EVG stains. And the idea is that you, with the machine learning, can pick up certain patterns that always kind of look similar in an unstable plaque in a symptomatic patient and less like this in a more stable asymptomatic patient. And then again, what this algorithm or this tool called iStar, which has been published by the Lee Lab in Nature Biotech last year, is kind of doing, it picks up these more or less hot zones in a plaque that always kind of look the same based on expression pattern within these lesions. And they are indicated here in red and in orange, that always kind of look very similar and kind of similar in an unstable plaque from a symptomatic patient. And then, of course, by using the spatial transcriptomics approach, you can look at what cell states are residing in these unstable areas, trying to understand, again, how they are crosstalking, and also what transcripts are involved in this destabilization in these recognized patterns based on this machine learning approach. And that brings me to the second part of the talk. So in the second part, I would more talk about the proteomics approaches that we've been doing in addition to transcriptomics. And there's been a lot of studies published, mainly looking at the proteomes of bulk plaques. So really just kind of analyzing plaques or pieces of the plaque without really knowing spatial distribution or spatial search hotspots that we are picking up, as mentioned, very similar to the spatial transcriptomics, where again, location really does matter in these patients. And what we've been doing to try to do a little bit different, and again, it's nothing we are doing in-house, it's something we're doing in collaboration with the Matthias Mann lab at the Max Planck Institute for Biochemistry in Munich, is to really do spatial mapping of these plaques and then to analyze certain subregions. And that's highlighted here. And I'll show some examples for that, where we've been microdissecting laser capturing certain areas of plaques, like the fibrous cap, the necrotic core, and also the media, and then compare that in between stable and unstable lesions to see differences and to get a more deeper insight into what's really driving plaque instability. And then more recently, and also show parts of that study, we've been doing this, what's called deep visual proteomics, where you can immunostain a certain cell type of interest, and you can again laser microdissect individual cells and then analyze the full proteome of individual cells, trying to understand also based on spatial location what different cell types are doing and how they contribute to plaque destabilization. And then more recently, and that's ongoing again based on this machine learning, is to determine these micromilieus, these microdomains that seem to be very relevant to plaque destabilization, identified with machine learning, and then to only look at these areas, again, in unstable and stable lesions or other clinical parameters, to again, understand the disease a little bit better and to essentially probably also look into serum and plasma from these patients to determine biomarkers that would help us to kind of predict or filter out more aggressive forms of atherosclerosis based on unstable lesion characteristics. But again, this first study is this kind of subregion study, or we term a subregion study, where we've been looking at 112, I think, stable and unstable plaques. They were kind of distributed into two groups, stable and unstable. They kind of had matching AHA criteria. They had the patient cohorts were very matched, so based on gender and risk factors that these patients had, really trying to only understand what's driving plaque biology and what's driving plaque instability. And then again, as mentioned, dissecting out different areas, the surrounding media, the necrotic core, and the fibrous cap, and then doing mass spectrometry analysis on these samples, filtering in a lot of the clinical metadata. And at the end, we also looked in the plasma from these patients, as well as an additional 1,000 patient samples, trying to build biomarker panels, kind of like a rule-in, rule-out biomarker to determine whether some patients have more aggressive forms of atherosclerosis that probably require more often imaging or other checkups, or if these patients seem to have a lesser invasive form of atherosclerosis, kind of ruling them out. And it's never, in our case at least, it's never really been only one biomarker. It's always been a panel of 6 to 7 biomarkers that kind of helped us to predict which patients also then had recurrent events during a 3-month follow-up period. And this is kind of the result. So what's been interesting is that, yes, when you look at the subregions, all of these subregions have very distinctly different proteomes based, or as indicated here in this PCA plot. And you can look at what proteins are different or specifically located to a certain subregion. And then by filtering in also the single cell data, you also know which cell type is likely expressing a certain protein. And as you would expect, so this is more confirmatory to the common knowledge that in particular in the media, you see a lot of smooth muscle cell developmental genes and intracellular programs that kind of maintain the healthy vessel wall. And the closer you move towards the necrotic core, the more inflammation, immune cell activation, and catabolism pathways are being turned on and more located to this necrotic core region of these advanced plaques. What's also been quite interesting is that if you filter in clinical variables, and that's been for us, as we've been mainly looking, to be honest, at the fibrous cap, which is kind of like the last line of defense. And we thought this is probably the most important structure of a plaque. But it was quite interesting to see that the necrotic core proteome really seems to determine what's driving an unstable plaque. And lesser the fibrous cap. Fibrous cap also important, but the necrotic core proteome was really the main determinator of an unstable lesion. And what's also been kind of interesting, but that's been shown before. Others don't really believe that, but that's something we've been seeing in Stockholm as well as in Munich, is that patients' symptoms don't really seem to drive changes in the proteome and cannot really help you to determine plaque biology and to really determine an unstable lesion from a stable lesion. And then again, of course, you can look at the different proteins being different. And as mentioned, most of the changes occur within the necrotic core. Interestingly, the top protein popping up is PCSK9, which of course is more known for its role in the liver and in lipid metabolism, but it's also very highly expressed, interestingly, in this more inflamed smooth muscle cell phenotype that also release it in the context of lesion destabilization. And some other proteins that have been shown here to seem to play a role in determining these unstable lesions. And again, what is kind of comes out on a more pathway-based GO-term analysis, unstable lesions, really the immune system plays a huge role, lipid handling plays a huge role, and in stability, it's really the maintenance protein and mRNA stability that seems to be driving a more stable phenotype. One step further and deeper into the plaque with proteomics technologies is this more novel approach that is called deep visual proteomics. That is basically single cell spatial proteomics, if you wish. What you have to do there, or what we've been doing here, again, in collaboration with Matthias Mann, is that we stain our plaques for ACTA2, so a marker of smooth muscle cells, again, just based on the knowledge that first of all, we have a lot of smooth muscle cells within the plaque. ACTA2 is a good antibody to determine them. And as we are interested in this changes in plasticity and this phenotypic switching that is occurring and how that contributes to lesion destabilization. So what you do is, again, in our case, we stained with ACTA2. You image these plaques, you segment them, again, based on a machine learning approach. They are being laser microdissected on an individual basis. And then after preparing the sample, they are being analyzed with high sensitive mass spectrometry. And in addition to that in situ study, looking at these human plaques, we've also been doing a study that we thought would help us to determine markers of these different smooth muscle cell subphenotypes or substates in vitro. So we've been using primary carotid artery smooth muscle cells and then driving them into different directions. So making them more foam cell-like, more inflamed, or more fibroblast-like, so more matrix producing by different stimuli that I'll introduce in a second. And then really to assess the cellular proteome, the secretome, again, based on mass spec. And also, and that's been quite interesting, to use glycoproteomics to look at the receptor landscape of these smooth muscle cells, because that's something that is for smooth muscle cells not really well determined. So what surface receptors they are being expressing, and that's something you can assess with glycoproteomics. Again, in our case, we've been driving them into different directions and then trying to assess how the receptor landscape is changing, which is very important if you're also thinking about therapeutic targets and how to get into a smooth muscle cell to understand the shift and the change they show with their receptors. And these are the different stimuli we've been using. So we've been using TGF-beta to make them more contractile again, PDGF-PP to make them more proliferative, more fibroblast-like, if you wish, free cholesterol to make them more foam cell, more inflamed, IL-1 beta, very strong stimulus to make them very inflammatory, and then this calcium phosphate-based osteogenic medium to make them more calcified or more osteogenic. And as you can see on this UMAP, all of these cells that you drive into different directions really seem to be very different from one another. And you can also look at different markers as shown here. So all the contractile gene markers like ACTA2 or transgelin, calponin, they all go up with TGF-beta treatment. If you make them more proliferative with PDGFB, markers like Ki67 and PCNA, which are usual markers of proliferation, are being induced with this osteogenic medium, markers like RUNX2, so really part of this more fibrotic, more calcification response, are being induced with IL-1 beta. You increase VCAM, ICAM, so markers of adhesion. And with free cholesterol, you drive them more into a foam cell type by increasing expression of LGLS3 as well as ABCA1. And then, as mentioned, parallel to that in vitro study, which helped us to determine markers that we can use to look at these different smooth muscle cell substates within these lesions, we've been using this TVP technology on ACTA2 positive cells. And what's been quite interesting already, when you look at the morphology of these cells, so in the media where you are expecting these more contractile smooth muscle cells, you already see that they have this classical like elongated shape that a smooth muscle cell, a contractile smooth muscle cell typically has. And the closer these smooth muscle cells are located towards the fibrous cap or the necrotic core, they already kind of adapt how they look. So they become this more cobblestone-like cell, which is classical for a phenotypically switched smooth muscle cell that has not this elongated shape anymore. It's not really exerting its contractile function anymore. And again, as mentioned, these cells are being segmented and then cut out on an individual basis. And you never really analyze, of course, that on a single cell level, but also as part of neighborhoods. So all of these neighborhoods are being analyzed then with mass spectrometry to understand changes in their phenotype and potentially also their function. And what's been quite interesting when we analyzed the data and we looked at the data is that all of these smooth muscle cells, irrespective of the location where they are within the plaque, they all seem to have this kind of core proteome, so kind of like a housekeeping proteome that doesn't really change, as well as this dynamic proteome that really changes based on the location where these smooth muscle cells are being located and eventually analyzed within the plaque. And again, as you would expect, so this core proteome shows a lot of housekeeping function, DNA repair, protein synthesis, gene expression and RNA processing. So all kind of really like basic functions that a cell has that are not exclusive to smooth muscle cells. But then in this dynamic proteome, you see all of a sudden then these terms like ECM, organization and remodeling, integrin signaling, immune response, very importantly, and also lipoprotein metabolism and transport. So again, giving you hints that within this dynamic proteomes, you see certain proteins that are very relevant and very important in this context of this phenotypic switch that these cells are undergoing in the athero context. And this is kind of how it looks then in situ. So ACTA2, so the more yellowish, light greenish, the higher the expression of ACTA2. And as you would expect, you see high expression of ACTA2 mainly in the media of these plaques. And as soon as these cells or neighborhoods can reside very close to the necrotic core or the lumen down here, they downregulate their contractile markers. So they lose their contractile function and turn and switch into something else. And one of the main things they are switching into is again into this more inflamed phenotype. So if you look, for example, at LGLS3, which is kind of like on the opposite side from ACTA2. So again, yellow meaning it's high expressed, and blue meaning it's there, but it's lowly expressed. So low expressed, you can see it or you can find it in smooth muscle cells that residing again in the media. But as soon as they move into the plaque shoulder or into the fibrous cap, they turn on LGLS3, which, as mentioned, is an important inflammatory marker and also important in foam cell formation. And if we've done this now for 24 plaques, kind of like as the main, looking at the main kind of transitions that these smooth muscle cells are kind of moving into. Again, ACTA2 being always highly expressed in the media of these plaques, but as soon as they migrate towards the cap or the core, ACTA2 becomes downregulated, and the signaling that becomes most significantly increased in these smooth muscle cells is interferon signaling, so inflammation. And this ABC family proteins mediated transport, so lipoprotein transport and handling. So these are the two main kind of, at least based on this pathway analysis or geoterm analysis, things that a smooth muscle cell kind of turns into once it's moving, becoming migratory and residing more closer to core and cap. And as a second cell type, so after SMCs, we also looked into other structural cells, fibroblasts, but also endothelial cells. And endothelial cells, as mentioned, was quite interesting for us because already based on the transcriptomic analysis, we saw, again, with very low resolution, that we have two very distinctly different endothelial cell phenotypes within our lesions. We have an EC1 and EC2 phenotype. And EC1, if we analyze this based on the single cell data and expression data, EC1 clearly seems to be more of an endo-MT kind of phenotype, whereas this EC2 is more of this angiogenic, more neovessel sprouting kind of endothelial cell phenotype. And the role of this has been, there has been some debate, especially about endo-MT, but I think for human disease, it's been shown that in human atherosclerosis, endo-MT is really contributing to fibrous cap thickening, preventing plaque rupture and stabilizing lesions, whereas angiogenesis has been shown, again, to be very relevant to trigger plaque inflammation and immune cell infiltration, because these angiogenic neovessels, again, when they become leaky, you have intraplaque hemorrhage within these plaques, and that's really the entry gate for immune cells into the human plaque, where much less you have this classical kind of transport over the lumen. So that's really how inflammation and inflammatory cells are getting into these human plaques. And again, we've been using deep visual proteomics to dissect out certain neighborhoods of these different endothelial cells that we are seeing. So we've been focused on mainly these luminal ECs, then these neovessel ECs, and finally this more medial endothelial cell phenotype. And then looking again at the full proteome of these individual cells and how they could potentially kind of contribute to plaque formation and plaque destabilization. And as you would expect, again, the luminal ECs indicated here in red are the ones that are more endo-MT like and more plaque stabilizing. Whereas in these neovessels, you see factors like complement activation, hypoxia signaling, so HIF1 alpha signaling, leukocyte adhesion, leukocyte chemotaxis. So again, kind of highlighting that these cells are heavily involved in immune cell trafficking as an entry gate into plaque inflammation and plaque destabilization. And this medial EC phenotype seems to be really this more classical, healthy endothelial cell phenotype that you would expect under less diseased condition, if you wish. So similar to the smooth muscle cells, we're also in this media region, we have the more contractile, more healthy state of these cells. And this brings me already to my conclusions. So I hope I was able to convince you that multi-omics is a neat and cool tool to identify novel processes of relevance to plaque vulnerability. That smooth muscle cells, maybe not the most important, but definitely highly abundant cell type in human atherosclerotic lesions, and that their profile and plasticity really differs based on the spatial location within the plaque. That link RNAs, long-encoding RNAs, are enriched in cells that show high levels of plasticity, like smooth muscle cells, but also endothelial cells, actually, where they seem to mediate that differentiation, or at least are involved in mediating differentiation into certain sub-cell states. Also, again, interesting, at least for us, very interesting that changes in the necrotic core really proteome seem to be the strongest driver and strongest determinator of plaque instability. So a lesser extent of fibrous cap, which to be honest, we expected in our lab. And then deep visual proteomics as a tool to really determine smooth muscle cell and endothelial proteomes in the future, hopefully also other cell types as well, and see how they, on a more single cell level and spatial location level, contribute to disease relevance. And with this, I would like to thank my lab, or my labs in Munich as well as in Stockholm. I have to highlight Ankit Zinya, who's from Toronto. So we have a Canadian in the lab. I promised him to say that. I'm not trying to recruit anybody, but it is possible. And all of our collaborators, all of our funding agencies, and of course, thank you for your attention. Thanks again for the kind invitation, Katie and Mireille. And I'll be happy to answer any questions. Thank you.
Excellent. Thank you so much, Lars. Any questions from the audience? I'm just monitoring online too. I have a question about sort of what you alluded to at the beginning about looking maybe to link what you're finding in the plaques with the serum and circulating biomarkers. And that's part of my question. And then related to that, what about sort of the genetics? You know, any links back to things like CHIP, things like SNPs that might be telling part of the story essentially of why, I mean, ultimately individuals have these different plaques. So why would be, some of these plaques belong to certain individuals and not others? And have you been able to make any connections?
Yeah, sure. So I am, well, so the first part with the tissue and serum proteomics, it's been challenging for sure to kind of see overlap. It definitely helped us quite a bit to do this sub-region study because again, all of this kind of panel of 6 to 7 proteins that we are seeing that I couldn't show today, but the papers also in revision, so hopefully soon, are coming out of the necrotic core. So and a lot of them, at least 2/3 of them, so 4 out of the 6 or 7, depends on how high you want the accuracy, relate to intraplaque hemorrhage. So intraplaque hemorrhage really seems to be a major driver of instability and a fairly good measurable marker of plaque vulnerability also in the serum samples. And I think that signal we would have likely missed if we only looked at the whole tissue sample. So I think the sub-region specific study definitely helped there quite a bit. So with CHIP, to be honest, we just started I think we have CHIP data from 500 patients now, and we have collaborators that have certain interests for a certain mutation, and then we kind of dig out the expression data that we have for these specific patients, and use that as a lookup strategy. So for us, it's more of a risk factor that we definitely want to have for our patients, but we've not been looking deeper into this. And then the genotype, yes, I think that's also super relevant. To be honest, also largely driven by industry collaborations. It's always the first question they have. I think I've just seen there was just been a comment in Nature that I think 80% of the FDA approved drugs from the last 3 years are bound to GWAS signal. And that's also and we have quite a few industry collaborations. That's one very important factor for them to know the genotype of the patients. And that's something we've been linking as well. And of course, that has a huge influence also on plaque morphology for sure. Yeah.
Okay, wonderful. Thank you, Lars, for this wonderful presentation. So I guess Katie stole my CHIP question, but I was interested in beyond the CHIP mutations and altering expression levels of genes within the tissue. Do you see maybe differences between CHIP associations with different cell types within the plaques?
Yeah. So yes, definitely. Again, we've been doing this more on a targeted approach, having questions. We have a collaboration, for example, with Alan Tall, who's very interested in JAK2. And yes, we know that the patients that have a mutation in JAK2, they have a very different fibroblast compartment, for example, interesting. So that composition and also the location of fibroblasts is very different in JAK2 mutated patients compared to some of the others. And we currently are doing another study looking at one of the DNMTA3 mutations, where the question is more driven by macrophages and whether there's a difference there. So yes, I think there's definitely a lot of differences that we've not really been exploring. But if you have a favorite CHIP gene that you're interested in, again, out of 500 patients, I think almost 40% have CHIP in our biobank.
Wow.
So
okay.
it's a lot of it's very
high.
present. But of course, I mean, this is an older patient, so it's like the average age 75. So you would expect kind of to see more CHIP. But I think whatever mutation you're interested in, we see it actually. At least from this more targeted panel approach. So it's super prominent, plays a huge role for sure. And we know also that some of the therapies are probably bound to CHIP. Canakinumab almost only works in some of the CHIP patients. So definitely very interesting. For us, it's been more, again, we are creating more these libraries right now, and CHIP is one of the books we are trying to put in.
Yeah, no, that's great.
Use and look up.
Okay.
Yeah.
And maybe one more question. For the link RNA you identified as being upregulated selectively in the modulated SMCs, DIX6, I think.
Yeah.
Is that regulated by KLF4, or do we know?
No, so it's also very interesting. So it's not signaling through KLF4. So DLX is one of the Drosophila homeobox kind of developmental genes. So it's known in humans or in any species to be super important in brain development. So the mouse is not viable because they don't develop a brain knockout for any of these three. So DLX5, 6, or the antisense. And for whatever reason, this gene or all of these three genes are being turned on in carotid plaques again. It's also very specific to carotid plaques. So it's not expressed in coronaries and peripheral artery disease. It's exclusive to the carotids. Might be the close proximity to the brain. I don't know. But it's interesting because, again, it's something that is kind of known to be shut off after brain development and then gets turned on in athero again. And there plays an important role. But the function is not through KLF4. So it's another mechanism that we are currently still, I have to admit, trying to explore. It's also not through DLX5 or 6, but there's other genes involved in this kind of switch into a more fibroblast-like cell.
Okay, fascinating. Thank you.
Hi, I'm Dr. Ducru. I'm a stroke neurologist at the Civic Hospital. So I don't know if you heard about like the recent publication of CREST-2, which is like make a lot of noise in the community because like these studies showed that like asymptomatic carotid stenosis, more than 70%, we should treat them with a stent.
Yeah.
So I'm just curious about your point of view and if you think, because like other stroke neurologists were kind of worried to send all our patients for stenting,
yeah,
especially when they have no symptoms.
Yeah.
So do you think there is any biomarker like which help in the future to select more of these patients?
Yeah. Yeah. So CREST-2 is something I've also been discussing, like I think with 10 different people right now. So I have to, yes, it definitely created a lot of buzz. So it's, I think, so, I mean, our surgeons also will not do that. So will not stent these asymptomatic patients. I think it is, I mean, in our cohort, 2/3 of the patients are asymptomatic actually that we are treating with surgery. So it's quite a high number, to be honest. And I think that's one of for us, the main motivation is to look for biomarkers in particular in these asymptomatic patients that could, again, give us like a rule in, rule out kind of theme to tell us, okay, this patient maybe doesn't need surgery, but you need to look at that. You need to kind of see him every 6 months and not every 2 years, which is then more of a rule out kind of thing. But it's challenging. I think it's difficult for carotid artery disease with the biomarkers because also it also relates to the symptoms. I mean, there's many more reasons beyond a carotid plaque that can cause a stroke, and that's also I think the difference to coronaries, where you always kind of know if you have an infarction in a certain area, you know what the culprit lesion, which coronary bed occurred. But for carotid artery disease, it's a lot more complicated. So that's very challenging, but it's something we are definitely thinking about, and I think so CREST-2 really helped us, I have to say, because I think the push for biomarkers is now bigger than ever. But it's a complicated disease. Yeah.
And maybe just one quick final question. We have a significant interest here at the Heart Institute in imaging, particularly developing sort of molecular imaging targets and tools. Do you foresee in sort of that, you said sort of plaque hemorrhage, necrotic core, proteome, like is anything bubbling up? I mean, we can talk later about some opportunities, but.
Yeah, so it's, we have 2 people in the lab that are working with our neuroradiologists to particularly look into this. And one of the ideas is to look for molecular probes for intraplaque hemorrhage. So I think that's something that's just based on all of our machine learning, all of our spatial analysis, really seems to be. And again, I can go on and talk about muscle cells forever, but I think intraplaque hemorrhage is super relevant, super important plaque stabilization, not only from an endothelial cell perspective, but also from an immune cell perspective, because that's really the entry gate into plaques. So if you can manipulate that either on the endothelial side or detect that on an imaging basis, I think it would be a super good strategy. So for us, it's really about the intraplaque hemorrhage and how to determine that.
Okay, wonderful. Thank you so much, Lars. Thank you for that excellent presentation, and thank you everybody for coming.
Thank you.